अस्सलाम वालेकुम एवरीवन। क्या हाल है सबका? हाउ इज एवरीबॉडी डूइंग? हेलो। अस्सलाम वालेकुम। चल एटलीस्ट कुछ तो रिस्पांस मुझे मिला। हाँ, कैसे है सब? व्हाट इशू बेटा? क्या मसला है मुझे बताओ, ताकि मैं इसको, इसीलिए लैपटॉप पर मोस्टली बच्चों को जूम ओपन नहीं हो रहा। अच्छा, कंप्लेन की है? मैं भी कॉल करती हूँ उसको। हेलो। हेलो। हेलो बेटा, स्टूडेंट्स कंप्लेन कर रहे हैं कि उनका जूम ओपन नहीं हो रहा है। हाँ, बच्चों का नहीं हो रहा। वो कह रहे हैं कि जी हमारा लैपटॉप के ऊपर से जूम ओपन नहीं हो रहा। एक किसी बच्चे ने लिखा कि मैंने मोबाइल पे लिखा, मोबाइल पे किया है तो फिर हो गया। जरा इसको देखो। हाँ, मैंने क्लास शुरू करनी बेटा। ठीक है, ओके। मैंने कंप्लेन की है बेटा, दे आर गोइंग टू लुक इनटू इट। तो जैसे ही ठीक होता है तो प्लीज काइंडली मुझे बताओ। और इन द मीन टाइम, जिस तरह से माहम ने किया है कि अपना मोबाइल ऐप ओपन कर लिए, आप लोग भी, बाकी लोग भी मोबाइल ऐप ओपन करके तो इसको ज्वाइन कर लो till it gets sorted out. और अब नेक्स्ट मुझे अपडेट दोगे? 45 पार्टिसिपेंट्स हो गए। नेक्स्ट अपडेट क्या है? Maham, anybody else? What's the update? ओके, आपको ये जिन जिन लोगों का तो जूम जो है वो या कनेक्टेड है, दे विल रीड दिस मैसेज। और जिनका नहीं हो रहा बेटा, उन लोगों को आप अपने तौर पे एसएमएस या मैसेज कर लो, या हुएवर इज द क्लास रेप, वो अपने ग्रुप के ऊपर मैसेज कर ले कि मूडल जो है उसमें कोई ना कोई टेक्निकल इशू है जिसकी वजह से वो आपके ब्राउजर या उसके ऊपर लैपटॉप पे ओपन नहीं हो रहा। तो दे आर सेइंग कि आप जहां जहां से आपका ओपन होता है, यू ट्राई ऑन योर मोबाइल वेयर एवर इट गेट्स वर्कड, तो आप उधर से उसको ओपन कर लो क्योंकि दे हैव अ टेक्निकल इशू। सो आप ट्राई करो, ताकि ये है कि आई जस्ट वेट फॉर अनदर 1 or 2 minutes, ताकि मैक्सिमम लोग जो है वो हमें, बिकॉज वी आर 72 पार्टिसिपेंट्स एट दिस मोमेंट, थोड़े बहुत और हमें ज्वाइन कर ले तो देन वी विल स्टार्ट विद द लेक्चर। So hopefully you can, all those who are online, you can hear me properly, वॉइस का कोई इशू नहीं है। हेलो। So everyone can hear me? आपको आवाज का तो कोई इशू नहीं है? Please let me know so that we can start with the lecture. आवाज आ रही है? Okay. चलो, let's start with the lecture. We are about the participants 75 तो okay. Let's hope the rest of you can join in as we progress with the lecture. So today we are going to start. Last time we finished with the parasitology. So today we are going to start another component आपका जो microbiology का component है, which is mycology. And again, as I told you previously, उसमें से एक क्वेश्चन आता है पैरासाइटोलॉजी में से, तो एक क्वेश्चन माइकोलॉजी में से भी आता है. इट्स अ कंपल्सरी क्वेश्चन एंड इट्स अ वेरी शॉर्ट टॉपिक. इसमें टोटल यू हैव 4 lectures in the book, and you do not need to go into you need to know everything there is, and but very simple questions come from this portion. Similarly, जब आपका वाइवा होता है उसके अंदर भी इसमें से a few questions जरूर पूछे जाते हैं, because this is a part of your curriculum, and definitely you get a question, you get questioned about it. So mycology is basically a study of the fungi, and if you, all of you are familiar with the fungi, mushrooms, and हमारी food, it is also used as a vegetable, and there are numerous thousands and thousands of species of fungi which are present in the environment. But इसमें से there are very few which are going to be pathogenic to man. So let's see what is mycology. Mycology is basically the study of fungi. It is made up of 2 words, myco, which means a fungus, and ology, which is the study of. So mycology is basically the study of fungi, and when we are talking about medical mycology, we are talking about those fungi which are going to cause a disease in human beings, or which are human pathogens. So fungal infections can also be called as mycosis. They can also be a intoxication of, because of the toxins produced by these fungal infections. They can be a disease or an infection in the body, and this is referred to as mycotoxicosis. Mycosis is basically any infection which occurs because of the presence of a fungus in the body, whereas mycotoxicosis is basically because of the toxins which are generated by those fungi, and when they are ingested, they are going to produce an infection in the body. So there are several thousand species, but less than 300 are pathogenic to man. This is most important. The kingdom is the fungi, and they are true karyotes. They are not as compared to the bacteria. The bacteria are the prokaryotes. These are the eukaryotes, just like the human cells. They have a true nucleus, but they are heterotrophic. Heterotrophic means that they need to have their nutrients from another. They are not parasitic. They feed on decaying matter, and some of them are parasitic, some of them are saprophytic, and but they require the nutrition from the environment, or from the environment where they are living. They cannot synthesize their own or their own nutrients, and they do not contain chlorophyll as compared to the plant cells. They produce filamentous structures. They can produce spores, and they are saprophytic when they are feeding on dead tissue, and they are parasitic when they are feeding on the living organism. All of them, they are going to require an organic source of carbon, and this source of carbon is generally obtained from the decaying food and vegetables and the plants which are present in the environment. This is a comparison of the fungi and the bacteria. There are a number of other comparative features, and there's a beautiful a table which is given in the book. So you can just remember the points which are present in that book for your SEQ questions. But basically, the a few important points are that the fungi, they are larger as compared to the bacteria. They are eukaryotic. They have a nucleus, whereas the bacteria are prokaryotic and they do not have a proper nucleus. The cytoplasm contains mitochondria and endoplasmic reticulum as compared to the bacteria, which is without these two. Then the cell membrane has a sterol, as well as as well as chitin, which is in comparison to peptidoglycan, which is present in the bacteria. Both sexual and asexual reproduction takes place, and there are different types of morphological forms of the fungi which represent their sexual proliferation and their asexual formation. But on the other hand, the bacteria do not require spore formation for their reproduction. Rather, the spore is just a method by means of which the bacteria require survival, or the bacteria, they remain in the environment under adverse conditions. Dimorphism is present in the fungi but not in the bacteria. And what is dimorphism? This is a phenomena in which the fungus is able to survive in two different morphological forms. We'll discuss that later on. Then the metabolism requires organic carbon, and they do not survive in anaerobic conditions. Whereas the bacteria, they do not require organic carbon, and they can make their own nutrients, and they may be aerobes as well as anaerobes. Different classifications of fungi have been given in different books. We have and they are dependent on different types of features which have been observed in these fungi, and they could be a morphological classification which is based on their different morphological features. There can be a systematic classification, that is, which or systemic classification, which is based on the system of identifying these fungi, or they can be a clinical classification which is based on what type of diseases they are going to produce. Most commonly, we classify the fungal infections or mycosis on the basis of their clinical classification, because we are dealing with the medical mycosis and we are dealing with the infections which are being produced by these organisms. So if it is going to infect the cutaneous, number one is the cutaneous mycosis. This is going to infect the superficial layers of the body. This would include the skin, the hair, and the nails, and it the deeper tissues are very rarely involved, and these are also known as the dermatophytes. Derma from the skin, and dermatophytes, the fungi which are infecting the skin layers. Then number two group is the subcutaneous, and this is confined to the subcutaneous tissue. Again, it may go into the little into the tissues, surrounding tissues, but it does not spread systematically. It does not go into the bloodstream, it does not go into the lymphatic channels, and does not go to the other organs. Causative agents are generally found in the soil, and whenever there is a trauma to the skin and there is a breach in the skin cavity, it is going to be introduced. If this is the fungus is present in the soil, or if its spores are present in the soil, they can be introduced into the body through that trauma, through that breach in the skin, and they can enter into the body and then produce a disease. But cutaneous, only the skin, hair, and nails, and subcutaneous is the subcutaneous tissue. Third group is the systemic mycosis. Systemic mycosis involves the skin and it involves the deep viscera. It becomes widely disseminated, and there are certain organs which are going to be specific for a particular fungus to produce an infection in. And last group is the opportunistic mycosis. Opportunistic mycosis are mostly the saprophytes. That is, they are living on the decaying matter, and they are not actually pathogenic to human beings. But if they gain entry into the human tissues, then they become pathogenic and invade the tissues in certain conditions, like immunocompromised patients and patients who have diabetes, cancer, leukemia, and these are the patients who are immunocompromised, and in them there the immune system is going to be very low. And in this situation, if the organisms, if these fungal pathogens, they gain entry, despite the fact that they are non-pathogenic, they may produce a disease in such individuals. Predisposing conditions, like if the patient is on steroid and antibiotic therapy, if the patient has a gamma globulinemia, that is the gamma globulin component is slow, is very less, then in all these conditions the body's immune system or body's defense mechanism is hampered, is not working properly. So these organisms, they can produce an infection, and these are known as opportunistic infections. The definition is generally the same as in case of the bacterial infections. Again, over there we have the pathogens which are not normally pathogenic, but if in immunocompromised individuals, if they gain entry into the body, they will be able to produce a disease. Clear? So you need to remember this class, the clinical classification, and just one classification. I think this is the classification which is given in the books, and this would and most of the questions, they come according to this classification in the exams. Any questions till this point? No questions? Mycology basically is going to be taught as a very basic and a very simple um, subject. So yes, you can just remember this classification, because I don't think so anything else is given in the book. Okay. Then we have a second classification, which is based upon the morphology of these organisms. So they can be divided into three broad groups. Number one are the filamentous fungi, or the molds, and these are the branching filaments which you can see in a when these organisms, they grow on a petri dish, or when you can for example at home, when your bread becomes stale, double roti jab kharab ho jati hai, to uske ऊपर green green या वाइट वाइट सा आपको छोटे छोटे से वो फंगस, या जिसको उर्दू में फफूंदी कहते हैं, वो नजर आने शुरू हो जाते हैं. ठीक है. एंड इफ यू आर गोइंग टू लुक at these, if you pick up a specimen and you are going to look at it under the microscope, you will see that it is made up of long branching filaments, and these filaments, they are known as mycelia. A single one is known as a mycelia, Mycelium, a single is known as a hyphae, and the collective mass of filaments is known as a mycelium or mycelium. And these branching filaments, they may be septate or non-septate. Septate का मतलब होता है एक division, ठीक है? अगर एक long tube है, hyphae are just like long tubes, ठीक है? And these long tubes, for example, आपके घर कोई भी पानी का पाइप है, कोई भी पाइप है, these are just like small, these long slender pipes of different sizes, ठीक है? अब उस पाइप के अंदर there are no सब डिविजन्स, कोई उसके अंदर डिवाइड नहीं हो के उसके खाने नहीं बने हुए। अगर तो वो डिवाइड नहीं हुआ और उसके खाने नहीं बने हुए तो देन इट इज नोन एज नॉन सेप्टेड। और अगर उसको बीच में उसके सेप्टम आया हुआ है या बीच में उसके डिविजन्स आई हुई है तो देन इट इज नोन एज सेप्टेड। सो on the basis of these septate और non septate, these are the morphological features जो कि classify करने के लिए और इनको ग्रुप्स में डालने के लिए these are the identifying points. Then second जो है वो yeast है। yeast कॉमनली आपने यीस्ट जो है वो हाउस होल्ड में देखा होगा एंड एवरीबडी नोज कि यीस्ट जो है दैट इज अ लिविंग ऑर्गेनिज्म। और उसको जब आप अपनी फ्लावर में डालते हैं विद शुगर एंड विद वाटर, विद वार्म वाटर, तो दैट बिकम्स एन एरिया वेयर इट कैन स्टार्ट ग्रोइंग। सो इट इज ए लिविंग ऑर्गेनिज्म which when it is provided with the proper nutrients, the carbon source and the warmth, that is the warm water and moisture, it will start reproducing and start growing. So and this is commonly seen, हम utilize करते हैं yeast बहुत सारे products में, but mostly we utilize it in bread, pizza बनाने के लिए and all these things. Okay, so yeast, what are these are basically rounder oval bodies and these reproduce by the formation of buds and these buds they are known as the blastospores. So एक जो round oval body है उसका एक छोटा सा protrusion निकलेगा और वो protrusion जो है that is known as a bud और फिर वो जब थोड़ा सा बड़ा हो जाएगा तो वो उससे disconnect हो जाएगा and then it is going to become an individual yeast form. And then third are the yeast-like fungi, and these are mostly which are going to cause disease in human beings, and they are formed because of these budding yeast cells. They remain attached to the original yeast, and this will give us the appearance of a broad septate hyphae, and they will it will feel like that they have a hyphae, but these are actually not the actual hyphae as we see in the filamentous molds. These are just the extensions. वो जो yeast से budding हो रही है वो budding जो है वो separate नहीं होती, और जब वो separate नहीं होती तो it becomes a part of the original yeast और वो फिर उसकी छोटी छोटी branches जो हैं वो उसके साथ ही attached होती हैं, and they also have a septum, and these are known as the pseudo-hyphae. Or this is a very important feature for the identification of Candida species. Candida जो है एक one of the species or one of the fungal species जो कि yeast form में होती है, और yeast form में it is a very common infection of the human beings or the human body. And ये infection mouth में भी हो सकती है, skin इसकी skin की भी हो सकती है, it can be of the UTI or it can be of the genital tract, and it is a very very common infection, especially in individuals जो diabetic है, especially in individuals who are on steroid therapy or on antibiotic therapy. So this is the form of yeast which we are talking about. This is the that morphological form. जब ये आप grow करते हो culture plates के ऊपर, तो this is what you can see, small pasty colored colonies, pasty off white colored colonies, और ये typical है कैंडिडा की इन्फेक्शन के लिए। और ये when you are going to see it under the microscope, so these are the yeast cells, ये जो individual हैं these are the yeast cells, ये उसकी budding है, ठीक है, and this is budding and this is the basically a feature which we see of the yeast cells, ये सारे के सारे, and if they have this budding, और ये budding जो है ये इससे separate नहीं होती, तो this portion is then known as a pseudo-hyphae, ठीक है, and this is again characteristic of Candida. ये on the other hand these are the molds, ये प्लेट के ऊपर ये ग्रो किया हुआ है, Aspergillus species है, ये एक और Penicillium species है जो कि ग्रो की हुई है, and then when you see it under the microscope तो आपको ये इस तरह के hyphae नजर आते हैं which are, और ये जो lines इसके अंदर नजर आ रही हैं these are the septum, these are the septum, and this septum indicates that this hyphae is a septate hyphae, ठीक है। तो सेप्टेट हाइफी कुछ स्पीशीज में होंगे, कुछ स्पीशीज में नॉन सेप्टेट हाइफी होंगे. तो ऑन द बेसिस ऑफ दिस we are going to identify, and this is the morphological classification. What you need to remember is that on the basis of morphology we can divide the fungi into three main groups. Number one are the molds, number two are the yeast, and number three are the yeast like fungi. और ये जो है this is again a pseudo hyphae. ये आपकी yeast cells हैं, yeast cells के बाद, and this is again the pseudo hyphae in case of Candida. Then on the basis of their reproductive modalities, yeah, on the basis of how they reproduce and what types of spores they produce, they are divided into four different groups. Number one is the on the basis on all of those which undergo a sexual reproduction by the joining of the nuclei and then the production of spores. They are known as the Ascomycota, the Basidiomycota, and the Zygomycota. And on the basis of asexual reproduction, that is, they are going to reproduce by means of budding. They are known as the mitosporic fungi or the fungi imperfecta, and these are mostly medically important fungi which are included in this group. They do not have a sexual reproduction, they just form buds, and they are going to reproduce by that methodology. So on the basis of sexual spores, we have these three groups: ascospores, basidiospores and the zygospore. अब ascospore जो है ये आपका एक sac के अंदर produce होता है, and it produces by this budding, and you have this smaller structure is known as an ascus, and this is a sac-like structure, और उसके अंदर 8 spores produce हो जाते हैं. And when these spores when this gets filled with these asci which contain 8 spores, तो ये जो structure है that is going to burst, and these spores they are going to be released. Then Basidiospores क्या है? Basidiospores again it's a sexual spore, और इसमें club like एक structure होता है on the end of the hyphae in case of certain fungi, and this is known as a basidium, and this is known as the basidium, and the spores which are produced at that end, they are known as the basidiospores. जबकि third sexual spore है, that is the zygospore, and this is a thick walled spore which is formed during the sexual reproduction. दो cell walls जो है ना वो fuse करके और उसके अंदर से वो एक spore produce होता है, and this is known as the zygospore. ठीक है, so you need to remember that there are three types of spores: ascospore, basidiospore and zygospore. Then when we talk about the asexual spores, they are more in number and they are of different varieties, but the most important are number one is the Chlamydia spore. ये hyphae है, और hyphae के दरमियान में within these hyphae these structures they start developing, and they start developing their spore like characteristics, and these are oval shaped, and they are going to be formed within the thick walled hyphae, and these thick walled hyphae, and this is characteristic of Candida species as well, और ये जो है these are known as the Chlamydospores. फिर उसके बाद आता है Sporangiospores. Sporangiospores जो है they are going to be numerous in number, and this is again the hyphae end, और hyphae end के ऊपर एक bulb produce होता है और इस या sac produce होता है, और sac के अंदर numerous spores जो हैं वो develop होते हैं, and these numerous spores once they are going to be formed, और ये जो hyphae है that is going to be found in the air. जैसे वो मैंने आपको mold दिखाया था, mold जो है that is something which is visible, that is made up of a large number of hyphae, और वो आपको जब examine करते हो under the microscope, so this is the type of a structure which you will see, और उसके अंदर फिर आपको ये एक sac नजर आता है, और sac के अंदर numerous spores, and this is known as the sporangiospore, और ये certain species में होते हैं like the Rhizopus mucor species of the fungi. Then we have the Conidiospores. Of Conidiospores जो हैं उसमें और Sporangiospore में difference ये है कि ये भी aerial hyphae के end के ऊपर होता है. This is also in the environment, and but it is not enclosed in a sac. इसका जो है वो tip के ऊपर they start producing spores, और वो spores जो हैं वो they remain in the form of a chain. and generally they are single, but single chains होती हैं और multiple chains बन जाती हैं at different ends of the tips of these hyphae. ठीक है, and so this is seen in case of Aspergillus or in case of Penicillium species. Then we have the Blastospores, which is in this case you have a bud at the end of the aerial hyphae like this. There is a bud, and उसके ऊपर आपको एक parent cell जो है ना वो नजर आने spores नजर आने शुरू हो जाएंगे, which are known as the blastospores. This is again seen in Canada albicans. And lastly you have the arthrospores. और arthrospores का क्या होता है? arthrospores इस तरह से बनते हैं कि ये आपका hyphae है, hyphae जो है वो disintegrate होने शुरू हो जाते हैं जहां जहां उनका septum होता है. These are the septate hyphae, and wherever there is a septum, that spore separates from the fungal mycelia. ठीक है, जो अगर टिप के ऊपर जो भी जितना भी सेप्टल एरिया है वो एक पूरा का पूरा स्पोर बना लेता है, and these are brick shaped structures, and these are known as the arthrospores, and this is seen in case of Trichosporum species. So we have three types of reproductive these three types of classifications. Number one was on the basis of the clinical manifestations, that is the tissues in which they are going to be producing a disease. And number that was the cutaneous, subcutaneous, systemic, and then the opportunistic. Second was on the basis of the morphological characteristics, that is they can be into yeasts, into molds, and then the yeast like fungi. And third was on the basis of the reproduction, those which undergo asexual reproduction and those which undergo in asexual reproduction. And most of the medically important fungi, they are going to be a part of the those which undergo in asexual reproduction, and they're also known as the fungi imperfecta. Any questions till this point? Any questions, beta? I ask you this so that I know that all of you are connected. Okay, so do answer these when I ask you if you have understood everything. It is just my way of asking you that all of you are there and I'm connected to you. Okay, so coming to the cell structure of the fungus, the cell wall is different from the cell wall of the bacteria, and it is consisting of two types of sugars or polysaccharides. Number one is the chitin, and the chitin we call it, and number two is the beta glucan. And both of them, they are polysaccharides, and this is also the site of action of some of the antifungal drugs similar to the bacteria. Then number two is the cell membrane, which is interior to the cell wall is the outermost component, and then when we go towards the inner side, we have the cell membrane, and this consists of ergosterol rather than cholesterol. In the bacterial cell wall, cell membrane, you have cholesterol, but in this you have the ergosterol. So this again becomes a site of action of the antifungal drugs, and it is going to attack the cell membrane of the fungus rather than the cell membrane of the other structures which are present in the human beings as well. So amphotericin B and azole group, they are the drugs which are going to affect the cell membrane of the fungi, and most fungi are the obligatory aerobes and others are facultative anaerobes. So as we talked about, there are no strict anaerobes in case of the fungi. The class of fungi is characterized by two forms of growth. Number one is now we come to the dimorphic fungi, and dimorphic fungi means that this is a fungus which can grow in two different forms. Di means two, morphic means morphology, and fungus is a fungus. So we have a fungus which is going to grow in two different forms at different temperatures. It grows as a mold with septate hyphae in the natural reservoir, that is the soil, or when it is incubated at 25 degrees centigrade on the fungal media in the lab. And this media could be separoid dextrose agar or potato dextrose agar, any media which supports the growth of the fungus. And it is when it is incubated at a temperature of 25 degrees centigrade, in that environment it will grow into a mold, and it has septate hyphae. And remember that the temperature for the bacteria is 37, but for the fungus it is 25 degrees centigrade. And number two if that same fungus, if it grows as a yeast in the tissues of an animal or person, or when it is incubated on the media, enriched media, and it is incubated at 37 degrees centigrade, it is going to also grow. So it grows both as a mold as well as a yeast under different circumstances and different temperatures, as a mold at 25 degrees centigrade and as a yeast at 37 degrees centigrade, as a mold in the environment, as a yeast in the human body. And it will also grow on the fungal media and at different temperatures. So this is the phenomena of dimorphism, or and these fungi are known as a dimorphic fungi. And this is again a very important SEQ question as well as a viva question. So in the humans, it is going to bud in the form of yeast cells, and in the environment it is going to produce hyphae and spores. So this is dimorphism, and if you remember this diagram, and you can just write 1 or 2 points over here, you will never forget the definition of a dimorphism. Then there are certain terms which you need to remember. Number one is the hyphae. This is a hyphae. This is a hyphae. This is a hyphae producing spores. And hyphae is a long branching filamentous cell, and they are the vegetative form of the fungal growth. And if they are numerous hyphae, then this is known as a mycelium. And this is an intertwined mass of hyphae, and that forms a mold colony. And this can be vegetative or aerial. Vegetative when they are growing into the soil, or when they are attached to any surface, for example onto the bread slices, onto any other bread or any other such things as food articles, or onto the plants, etc. And the aerial that grow up from the surface, and the aerial is the point, is the end where you will produce these spores. Pathogenesis, basically it's the transmission, then the colonization and infection, and the end organ dissemination. So basically all those which are systemic in nature, they are going to colonize, they are going to produce the end organ dissemination. So the transmission can be from from vertical, from the mother to the fetus, to the child during the vaginal delivery, if the genital tract is colonized with the fungi. So it's very likely that if the mother is going to have a fungal infection of the genital tract, it can be transmitted to the child during the delivery. Then there can be a horizontal transmission from patient to patient, from contaminated services, healthcare workers. They can all transmit this infection to the other individuals. Then there is the colonization of various structures in the body. It could be the skin, the respiratory tract, GIT, or the catheters in case of seriously ill patients. Then the colonization is going to depend upon certain patient factors, organism factors, and the or certain medications if the patient is taking. So the patient factors include an immune defense mechanism is immature as it occurs in the child, or if they are immunocompromised. Then if the skin surface remains very moist, warm, and that is a very good soil for the growth of the fungi. Then if there is a trauma to the skin, there is going to be a fungal dermatitis. There can be other if there is an intestinal perforation, and the perforation is towards the outside, and that can also result in the infection with the fungi. Then any abdominal surgery, hyperglycemia, all of these are the predisposing factors. Intestinal perforation, abdominal surgery, hyperglycemia, invasive catheters, tubes, all of these are going to predispose to the colonization of these fungi. Then there are the organism factors. That is the fungi which have more virulence properties. The greater the number of the organisms, the greater the multiple site colonization and adherence. Then if the patient is on antibiotics, if the patient is on certain parenteral nutritions, they are going to take certain steroids. All of these, they are also going to be a factor which are going to promote the colonization of the fungi in the body. Then we have the Types of infections. Once it colonization has taken place, then because of the immature immune defenses, or if there is any other co-infection, these are able to produce an infection and be carried by the blood, by the urine, CSF, peritoneal fluid. All of them, they can be infected by by these fungi, and they can produce a dissemination of these organisms to the different parts of the body, resulting in involvement of the heart, of the kidney, of the central nervous system. Fungal meningitis is a very common etiology. Then liver abscess can occur, cutaneous abscesses, etc. So basically, again, there is a transmission, there is a colonization, there is an infection and end organ dissemination. And when you study the pathogenesis of the different groups of fungi, you will understand this more clearly. Fungal infection, basically it forms granulomas. For example, in case of histoplasmosis and blastomycosis, both of them, they are the systemic fungal infections. Then a cell-mediated immune response is involved. Also, there can be an acute suppuration, that is the abscess formation, if there is infection with two types of fungi, that is the aspergillosis, which is present in the environment, and sporotrichosis, which again is present in the environment. Sporotrichosis is a fungal infection which is generally present on the soil, and it is also present on the plants, etc. And a common source of this spores getting into the human being is the prick from a thorn. For example, जो आपका गुलाब का पौधा है, उसके अंदर वो छोटे thorns होते हैं, या वो कांटे होते हैं. This can be infected with the sporotrichium fungus, और अगर वो infected है और वो कांटा आपको चुभ गया, तो it can be a cause of fungal infection in your finger. ठीक है. So उस सिचुएशन में वहां पे एक abscess formation हो सकती है. They do not have any endotoxin in their cell wall, unlike the bacteria, and they do not produce any an exotoxin, again unlike the bacteria. If you carry out the skin test for the exposure to the fungal antigens, then the cell-mediated immune response is going to indicate a positive skin test, indicating that there is a fungal infection. It does not indicate a current infection, because that exposure may have occurred in the past, and once that infection is there and a cell-mediated immune response has occurred, it is going to persist for life. The negative skin test, on the other hand, does make the diagnosis unlikely. So if you have a negative skin test to a fungal antigen, it generally rules out a fungal infection, except for the patients who are immunocompromised. Intact skin is a host defense against both the candida as well as the dermatophytes, because the fatty acid which is present in the skin, it inhibits their growth, and the hormones also which are associated with the skin, they also limit the fungal skin infections. As our body goes through the different changes, as it reaches puberty and goes forward, there are certain hormones which are going to be produced on our skin surfaces. They also limit the fungal skin infections, and the fatty acids which are already present, they also limit the fungal infections. And then we have the normal flora of the skin and the mucous membranes. Again, they do not allow the fungi to grow in that area. But if that normal flora is inhibited by antibiotics, then an overgrowth of these infections occur, and the most common of them are the candidal infections or the dermatophytes. In the respiratory tract, the host defenses are the mucous membranes of the nasopharynx as well as the alveolar macrophages. Both of them, they are going to fight against these fungal pathogens. Immunoglobulin G and immunoglobulin M are produced as a part of the immune response to these infections, but they are not protective in nature. So the body cannot utilize them to fight off the fungi. Cell-mediated immunity is basically protective, and if you suppress the cell-mediated immunity in an individual due to steroid therapy, due to any other reason, to any disease, for example, diabetes, cancer, chemotherapy, all of this is going to lead to the opportunistic fungal infections. That is why if you read anywhere, you will see that patients who are getting chemotherapy, who are diabetic, who have cancer, all of them, they are more prone to fungal infections as compared to the normal population. Three types of fungal diseases are produced. Number one is the mycotoxicosis, which is caused by the ingestion of the toxins. अच्छा, till this time we have been studying the fungal diseases, जैसे मैंने आपको बताया कि स्किन में है, subcutaneous tissue में है, systemic है, and that is because of the presence of the fungus in or over our body, on our skin surfaces or within our system. So those are the fungal infections. Then there are certain infections produced by the fungus, of certain diseases produced by the fungus, which are going to be the because of the toxins and the antigens which are present in the fungus. They are not because of the presence of the fungus in the body, rather because of the presence of the toxin or the antigen of that fungus, which is going to produce the disease. These are the mycotoxicosis, which is caused by the ingested toxin, and these are the allergies to the fungal spores in certain mycotic infections. Mycotoxicosis is basically produced by the mushrooms, eating of certain mushrooms. There are certain mushrooms which are poisonous in nature, and that is why it is suggested that if you are going into for a wild trek, कोई hiking पे जा रहे हैं, तो do not eat anything which seems like a fungus to you. जैसे हम लोग fungus basically if you must have आप लोगों को पता होगा कि वो छोटे छोटे mushrooms हमें tins में भी मिलते हैं, बाजार में भी हमें mushrooms जो हैं वो नजर आते हैं, and we do use them in cooking, और चाइनीज में तो स्पेशली बहुत ज्यादा यूज होते हैं चाइनीज कुकिंग के अंदर. बट दोज आर दोज फंगाई व्हिच हैव been identified, और जो wild में grow कर रहे थे, some of them are toxic. So they produce 5 types of toxins, and 2 of them they are hepatotoxic, that is they are going to affect the liver. और the most important इसमें जो है ना वो है, एक तो है ergotamine and LSD or lysergic acid diethylamide. ठीक है, this is produced by the molds and this is going to infect the grains. अब grains कौन कौन सी हैं आपकी? grains basically आपकी rice भी हो सकती है, but mostly it is the wheat which gets affected by these molds. ठीक है, और जब ये molds in wheat को इसको affect करते हैं, तो ये दो enzymes produce या दो toxins produce करते हैं, and if the wheat is infected with this and if we consume that wheat, it can cause a disease which is referred to as ergotism, and basic effect is toxin ka hota on the vascular channels and on the CNS. So different vascular and neurological effects would be produced by the ingestion of ergotamine and lysergic acid diethylamide, which is produced by certain molds. एंड जैसे कहा कि दे आर ऑन द ग्रेन्स, जिसमें वीट भी हो सकती है, बार्ली भी हो सकती है, अदर ग्रेन्स ऑफ दैट सॉर्ट. एंड सेकंड इज द एफ्लाटॉक्सिन्स. ये एफ्ला है बेटा, टी नहीं है यहाँ पे, ये AFLATOXINS है. Aflatoxins produced by the fungus which is known as Aspergillus flavus, and when this is ingested, they are going to cause liver damage and it can also cause hepatic carcinoma. So very common MCQ question is the association of which toxin with hepatic carcinoma, which fungal toxin is associated with hepatic carcinoma, and ये aflatoxins होते हैं. They are also going to spoil the grains and peanuts, और अगर उसके ऊपर ये toxin लगा हुआ है, तो then and if that is ingested, it is going to produce a fungal a toxic infection. Then allergies to this fungal spores are also very common, and if they are going to be inhaled, they can cause asthma. And spores of Aspergillus are very, very common for this. Aspergillus is very common in the environment as well. Plants इसके जो इसके spores हैं, जैसे मैंने उसको आपको दिखाया था कि they are in a sac and they are numerous in number, और जब ये sac break करता है, तो they are then dispersed into the environment and they remain in the air, and if they are going to be inhaled, तो उसके अंदर से lungs में जाके it can cause bronchoconstriction because it is an allergic phenomenon, और IgE produce होता है and IgE bronchoconstriction produce करता है, फिर eosinophilia भी होगा, and end result is going to be a difficulty in breathing, and the patient may develop asthma because of the spores of Aspergillus, and a skin test would be positive in such cases. Coming to the laboratory diagnosis. Laboratory diagnosis में आपके 4 main mechanisms हैं for the lab diagnosis of your fungal infections. Number 1 is the direct microscopic examination. Number 2 is the culture. Number 3 is the DNA probe test. And number 4 is the serological test. First of all, a few words about the direct microscopy. Direct microscopy में generally what we do is we use the light microscope as you have in your labs, and we are going to take a glass slide. We put the tissue on the glass slide, and then we are going to observe it under the microscope. Generally, we treat the tissue with 10% potassium hydroxide. 10% potassium hydroxide का function ये है कि वो skin के cells को जो 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 उस slide में होंगे, because स्लाइड में होंगे वो उनको डिसॉल्व कर देता है. सो इट डज नॉट हैव एनी इफेक्ट ऑन द हाइफी, इट डज नॉट हैव एनी इफेक्ट ऑन द स्पोर्स, एंड इट डज नॉट हैव एनी इफेक्ट ऑन द यीस्ट. सो treated with 10% KOH, तो जो स्किन सेल्स उसके साथ उस टिश्यू के उसके साथ आए हुए, स्पेसिमेन के साथ आए हुए, या एनी अदर cells which may be present when taking a sample, they get dissolved and you will just be left with the fungal elements if they are present. ठीक है. सेकेंडली यू कैन यूज स्पेशल स्टेन्स. स्पेशल स्टेन्स में आपका एग्जांपल है, फॉर एग्जांपल इंडिया इंक है. इंडिया इंक इज जस्ट लाइक नॉर्मल ब्लू इंक जो हम पेन्स में डालते होते हैं. ठीक है, इट्स अ ब्लैक कलर्ड इंक, बट इसके अंदर कुछ थोड़े से पार्टिकल्स होते हैं. सो Cryptococcus जो है, that is a fungus which has a capsule. So when we make a slide with the India ink, what we see is that we see a rounded structures, and the rest of the area is covered with the black ink. So these fungal structures, they become visible very easily. Second, which we use is the calcifluor white, which is a fluorescent dye, and that binds to the fungal cells and it can be utilized to identify the specimens. I'll just show you. दिस इज अ वेट प्रिपरेशन. ठीक है, ये हमने फंगल हाइफी देखे, और ये वाले जो है ये अंडर द माइक्रोस्कोप. इसमें हमने सिर्फ एक स्क्रैपिंग ली फ्रॉम द स्किन, या जिस भी एरिया में हम फंगल इन्फेक्शन सस्पेक्ट कर रहे हैं वहां से हमने स्क्रैपिंग ली, और स्क्रैपिंग लेके हमने उसको under the microscope देखना शुरू कर दिया with KOH, and this is what we will see. So it becomes a very easy method for the diagnosis. Secondly, अगर हमने stain करना है तो we can use lactophenol blue. Lactophenol blue एक dye है which is going to be picked up by the fungal structures, और फिर ये fungal structures against a white background आपको नजर आने शुरू हो जाते हैं. And then we have the calcofluor white जो कि एक fluorescent dye है. Fluorescent dye जो है that will get attached to these fungal structures, और फिर उसको अंडर द फ्लोरेसेंट माइक्रोस्कोप जब आप देखते हैं तो इट विल कम वेरी इजीली विजिबल. ठीक है, और इसमें ये है कि इन बोथ दीज केसेस यू विल सी, बिकॉज ऑफ द कंट्रास्ट, यू विल पिक अप द हाइफी एंड द स्पोर्स वेरी वेरी easily. In this case, you need to look, you need to spend more time, and you need to be more careful, because ये सारे के सारे बैकग्राउंड में मर्ज हो रहे होते. So this makes the diagnosis very easy. और ये है इंडिया इंक प्रेपरेशन. इंडिया इंक प्रेपरेशन में आपने सैंपल लिया, सैंपल जो है स्लाइड पे डाला, और उसके बाद आपने एक ड्रॉप इंडिया इंक का उसके ऊपर डाल दिया. जब आपने इसको अंडर द माइक्रोस्कोप देखा तो जो इंक है, दिस इज द इंक, दिस इज द ब्लैक बैकग्राउंड, और ये क्रिप्टोकॉकस है, और ये इसका सेल है, और ये इसका कैप्सूल है. So they have huge capsules, और ये इंक इनको penetrate नहीं करती. So these rounded structures, they can be very easily seen, और ये most commonly cryptococcus जो है वो cryptococcal meningitis produce करता है, and this test is carried out onto the CSF. तो CSF का sample लेते हैं, एक ड्रॉप उसका डालते हैं, एक ड्रॉप इंडिया इंक का डालते हैं, और उसके बाद उसको माइक्रोस्कोप के नीचे जब देखना शुरू करते हैं, so if you can see these structures, तो cryptococcus की identification हो जाती है. And then second diagnostic methodology is the culture के लिए, just like the bacteria, we have different media, and you just need to remember कि जो commonest media है, that is the Sabouraud's agar, and this Sabouraud agar की कुछ characteristics है which allows it to allows it to maintain the growth of the fungi, जिसके अंदर कुछ antibiotics है, and then it has a low pH because the fungus, they grow at a pH of 4 to 6 as compared to the bacteria which grows around at a neutral pH. ठीक है, और इसके अंदर ये yeast forms को भी support करता है, और ये mold forms को भी support करता है. And over here you can see the mycelia, and when we are going to take up a portion of this or this, and we are going to observe it under the microscope, तो हमें वो characteristic उसके features नजर आ जाते हैं, morphological features, which will help us in the identification of the particular fungus. thirdly, DNA probe test. अब ये colonies जो हैं, जो अगर हमें नजर आ रही हैं, before they can be seen by the naked eye, we can utilize them. जैसे जैसे हमें इसको let's go back. अब ये colony तो जो है, that is, fungus is slow growing, so it is going to take at least 7 to 10 days मैक्सिमम 14 डेज तक हम फंगल ग्रोथ करते हैं, ठीक है। सो कल्चर जो है, आई फॉरगॉट टू टेल यू, कल्चर जो है दैट इज केप्ट फॉर 14 डेज बिफोर इट इज सेड दैट इट इज नेगेटिव। सो जनरली ग्रोथ जो है वो 6 to 7th डे पे नजर आनी शुरू हो जाती है, एंड इट इज गोइंग टू बी केप्ट एट अ टेम्परेचर ऑफ 25 डिग्री सेंटीग्रेड। सो इंक्यूबेटर में नहीं रखते इसको, हम ओपन उसमें किसी इंक्यूबेटर में रखते हैं जिसमें टेम्परेचर 25 डिग्री सेंटीग्रेड पे मेंटेन्ड है, और if during the or if the lab temperature is at 25 degrees centigrade, you can also put it on a on the cabinet in the biosafety, in the biosafety cabinet outside instead of in the incubator, okay. So but this takes about, as I said, 6 to 7 days to appear. Yeast जल्दी appear हो जाती है, molds जो हैं they are going to take a longer time. So in that situation you can carry out the DNA probe test because they will identify them before you can see them visually, and the diagnosis can be made more rapidly. So we have DNA tests which are going to be which are basically but for the systemic infections like histoplasmosis, blastomycosis, and cryptococcus. So these, all those fungi which are going to cause systemic infections, we have the DNA test for them and they can be identified with that. Fourth is the serological test. Serological tests are going to again be helpful in diagnosing the systemic mycosis because the immune response is going to be triggered only when the organism reaches within the human body, and there is a rise in the antibody titer, but IgM and IgG both can be detected, but a single reading is not going to be effective or not going to be the deciding factor. A rise in the antibody titer is necessary to confirm the diagnosis because we are exposed to these fungal antigens and throughout our life, and we may have, and most of us will have IgG and IgM against these fungal antigens. So we need to have a rise in the antibody titer before we can confirm the diagnosis. Then a complement fixation test can be utilized. Then the latest agglutination test can be utilized, especially in cryptococcal meningitis. Skin test, they will detect the delayed hypersensitivity. It's a delayed hypersensitivity test, and they will detect the reaction to the fungal antigens that is injected intradermally. And if there is an exposure, you will develop this wheal and flare. But if there is no exposure, then there will be no such reaction on the surface of the skin. Fluorescent antibody test can be carried out. Again, the fluorescent dye is attached to added to the scrapings, to the skin scrapings or the tissues which you have taken, and when you examine them under the microscope, this is what you are going to see. Then if there is a tissue which is involved, then that is going to be again pathology की यहाँ पे spelling mistake है. अच्छा, again क्या कहते हैं, when if you have a biopsy, if some deeper tissues they have been involved, then you can take a biopsy and you can carry out the H&D staining or the hematoxylin eosin staining and the processing of the sample and examined under the microscope. And these structures, these long structures, Y-shaped structure, and these pinkish longish structures, these are the hyphae of the fungi, and these are the spores which you can see. Similarly, over here, you can see this. You can see these spores. These are the hyphae which are there, and these black structures, these are the you can see the budding over here, small yeast-like forms. So this is on biopsy and histopathology. A few words about the antifungal therapy. They are like the bacteria. This antifungal drugs, they're going to target different structures of the fungal cell. They can inhibit the cell wall synthesis. They can inhibit the nucleic acid synthesis. They can inhibit the ergosterol synthesis in the cell membrane, or they can interfere with the membrane function. So these are the four main groups of antifungal drugs, and I'm not going to discuss them in detail. The reason being, these are going to be, you must have been taught them in pharmacology, and they are you will have the details over there, and some of the content of the antifungal drugs, it is also given in your book, and you can just go through the table which is present over there, so that you know the names of the antifungal drugs if it is coming up in a question. But generally, antifungal drugs, they are not asked in a in context with the mycology question in pathology. So any questions? Okay, dimorphism, you want to get okay. Dimorphism is basically, let's see, if you have any fungal any fungus, okay, we will take the example of histoplasmosis. They are a number of fungi which are dimorphic in nature. We call those fungi dimorphic, which can grow both at 37 degrees centigrade and in the environment at a temperature of less than 30 degrees centigrade. When that fungus goes into a human body, that is the time when it is going to grow at 37 degrees centigrade, and it is going to grow as a yeast cell. Okay? On the other hand, if it is outside in the environment, it is still able to grow at temperatures of less than 37 degrees centigrade, and mostly at temperatures of 25 degrees centigrade. But in this case, its morphological form would be in the form of hyphae and spores. Okay, these spores, when they enter a human body, they will grow in the form of yeast cells. When these yeast cells, they go into the environment, they will grow in the form of hyphae and spores. Okay? So this is the difference between uh, this is the actual definition of the dimorphism. Any other questions? If you remember this diagram, you will remember what is dimorphism, because there are certain species like the histoplasmosis and blastomycosis, and basically which cause systemic infections. Those are the fungi which are going to be dimorphic in nature. And if the only thing which we need to remember is that within the humans, they're going to grow as yeast cells, and within the environment, they're going to grow as hyphae and as spores. Any other questions? No questions? Understood? Okay, thank you so much. Just go through this chapter. Just give it a reading. I'm not asking you to remember the chapter. I'm just asking you to give it a reading so that you are familiar. It's just 1 or 2 pages, 2 pages. So just give it a reading as if you are reading a storybook, and you will understand what has been taught to you, and then the difficulties will not be there when you start learning this chapter. Thank you so much. Stay safe. Take care of yourself.